Hey everyone, Mr. Walker here. We finished off the last presentation on neuron function with the message traveling down the length of the axon that I'm drawing on the board here, reaching the axon terminal or synaptic knob. And just drawing the rest of the axon here, the cell body, and remember that coming out from the other end of the cell body where the information is being received, we would have the dendrites. So if this is a motor neuron that I'm drawing, that means that the motor neuron would have to be receiving the information from the central nervous system. And of course, it's the dendrites that are going to be picking up that information. And then this information is going to be coming from interneurons within the central nervous system. So I'm going to draw in a whole bunch of interneurons here, and I'm going to really simplify it, just draw the axon terminal of several interneurons, and a few of them here, I'm just going to use a green line, green marker, and again, this is just showing the axon terminal that would be coming from many different interneurons. And for any given motor neuron in the central nervous system, it's not going to be just one, but it will be dozens or even hundreds of interneurons that are trying to tell it what to do. Now, when I say trying to tell this motor neuron what to do, there are one of two different possibilities. What I've drawn on the board right now with the green lines, this is going to represent an interneuron that's going to lead to depolarization in the dendrite end of the motor neuron. And because it leads to depolarization, because it can lead to an action potential being generated and passed down the length of the motor neuron, this is referred to as excitatory. So the green line's excitatory interneurons that are going to lead to depolarization and possibly the generation of an action potential. We can also have exactly the opposite though. There are also interneurons within the central nervous system that are trying to tell this motor neuron to do exactly the opposite, which is not generate an action potential. And in fact, instead of leading to depolarization, they would lead to the opposite, which is hyperpolarization. And that, in fact, would make it more difficult for this motor neuron to generate an action potential. So these lines here, the red ones, they are inhibitory. Oops. So you can also think of these excitatory ones as kind of the go signal, telling the motor neuron to go ahead and send a message, and the inhibitory ones as a stop signal saying don't generate and send an action potential. At the other end, we do have an axon terminal, of course, and what we would generally have there is another cell. So the other cell could be, well, possibly another neuron. If this is a motor neuron, it's probably a muscle, and this is usually how a muscle is drawn and represented. It could also be a gland as well. Now, one of the things that I do want you to notice is that, well, what I try to draw anyway, is that there actually isn't a physical connection between the axon terminal of one neuron and the dendrite of the other neuron, or in the case of the muscle, between the axon terminal and the muscle. There is a space. So everything that we've talked about so far, the action potential and how the action potential is passed on through the dendrites, the cell body, and the axon itself, won't work for passing the message on to the next adjacent cell. So we need a different process here. This picture here is showing, well, it looks like one single dendrite in the background, and for this one single dendrite, what we have again is many other neurons that are trying to tell it what to do. So maybe these ones that I've circled in green are excitatory ones, and perhaps these ones here that I'm circling in red, perhaps those are the inhibitory interneurons. But a whole bunch of them that are trying to influence this one neuron in terms of whether it's going to send a message or not send a message. So a bunch of new terminology here for what is going on with this connection between two different cells. So a junction, the junction itself, which comprises the ending of one neuron or the axon terminal, and the sending neuron of the sending neuron and the dendrites of the receiving neuron. The term that you need to know for this is a synapse. There are some different kinds of synapses. There are electrical synapses, there are chemical synapses. The one that we're going to focus on, it is what is referred to as a chemical synapse, and it does involve the generation and the release of a chemical from one neuron that's then sent and detected by another neuron or another cell type. So the picture that we're taking a look at here to the right, we have two cells at the top. Cell number 1 in this case is going to be the neuron that is sending the message. So the message in this case is going to be going from the left-hand side through the dendrites, the cell body, the axon, eventually reaching the axon terminal. Cell number 1, this one here, is the one that's going to be receiving the message. So that's the receiving neuron. Now if we take a look at what's actually going on between these neurons, that's the enlarged view that's at the bottom, and a number of different structures here that you need to be able to identify on a diagram. You should be able to draw a diagram that is showing the synapse with all of these different structures and know the function of these different structures. So if we take a look at number 1, I did say that that is the receiving cell. There is a little bit of a more technical name for it. It is referred to as the post-synaptic neuron, post meaning after, at the latter half of the synapse. That would be the receiving cell. The other cell, number 2, the sending neuron, is the one before the synapse, so it is called the presynaptic neuron. In order to pass the message on from one neuron to another cell, what we do need to have are some chemicals. These chemicals are housed inside of tiny little vesicles or sacs that you do find in the axon terminal, and these are referred to as synaptic vesicles. So number 3 on the diagram are these chemical-filled synaptic vesicles. What they really are is just a small phospholipid bilayer in the form of a sphere, and inside of the sphere is where you do find these chemicals. Number 4, hopefully you recognize number 4. Number 4 is the mitochondria. This is a very energy intensive process, and of course mitochondria are responsible for the generation of ATP, the universal energy source within the cells. Quite often the term synapse is used incorrectly. As I have here, a synapse is the junction between two cells. It does involve the end of one cell, the beginning of the second cell. When we talk about the actual space between the two of them, it is given a different name. It is the synaptic cleft. So the synapse involves several different structures. The synaptic cleft is specifically the space between the two cells. The chemical that's going to be transmitting this neural message does have a more specific name, and it is called a neurotransmitter. So the chemical is a neurotransmitter because it is being released by a neuron, and it's going to be diffusing across this very, very small space, the synaptic cleft, and it will target and influence another cell. In this case, it's another neuron that's being influenced. And that other neuron is the postsynaptic neuron. So the last thing that it has labeled here, number 7, is the postsynaptic membrane, the cell membrane of the receiving cell, or the postsynaptic neuron. Picture here in the background is a very, very enlarged, magnified electron micrograph picture showing the junction between the axon terminal or synaptic knob and the dendrite. So where we have labeled A, that region is going to be the axon terminal, and all of these tiny little circles that we see here, those are not neurotransmitters, those are the synaptic vesicles. And in fact, inside every single one of these synaptic vesicles, there could be hundreds or thousands of individual neurotransmitters. Down toward the bottom, labeled with a D, this is going to be the dendrite. And if you take a look at these arrows right here, what they're pointing to is what's really difficult to see, but there is a space here. And it is a tiny, tiny little space between these two cells, and that is again the synaptic cleft. The picture that I have at the upper right-hand side, very difficult to read what the text is here, but what it does show is that we do have some neurotransmitters that are being released from the axon terminal at the top. They're diffusing across the synaptic cleft and they're binding to what is called a receptor. What this picture is showing, and what they talked about in this article from Scientific American, is the process of learning. And what happens through the process of learning is that you're forming stronger and stronger connections between these two cell types. And this right here that I've circled in black, they're called adhesion molecules. As you learn, as you go through a repetitive process of studying and rote learning, what you're doing is you're strengthening the connections between neurons in the brain, and that is what they think is taking place through the learning process. So a couple of nice pictures that I'll show you on the next two slides here that are showing the events at the synapse, and you should be able to take a look at a picture like this and you should be able to label the different structures that we have here. So at the top, what we do have is the presynaptic neuron and the presynaptic membrane. This is the axon terminal. Here it says the axon of the sending neuron. It is the axon terminal that it's showing here. All of these circles, they are the synaptic vesicles, and the green circles inside of each synaptic vesicle, those are representing the individual neurotransmitters. Now we have this space, and remember the space is referred to as the synaptic cleft. So these chemicals, when they are released from the sending neuron, the presynaptic neuron, they do have to make it across this space, and the process of diffusion is really only efficient over very, very small spaces, and that's why this space has to be kept to a minimum. At the other end, we do have the receiving cell, or the post-synaptic neuron, and what is going to be receiving the message are going to be receptors, chemical receptors that are very, very specific for one and only one neurotransmitter. And what we'll see is that the neurotransmitter is going to attach to these receptors, and when it does, it's going to lead to a response in the receiving cell. And that response could be really one of two different possibilities. It could be excitatory, causing depolarization, or inhibitory, causing hyperpolarization. So let's take a look at this animation.
Neurons communicate across junctions called synapses. When an action potential arrives at the end of an axon, a signal is transmitted across a synapse to a receiving cell. Although synapses may be either chemical or electrical, we will only look at a chemical synapse in detail. How do signals travel across a chemical synapse?
There is a narrow gap, the synaptic cleft, between the synaptic terminal of a sending neuron and the surface of a receiving cell. An action potential arrives at the end of the axon of the sending cell. This causes chemical changes that make vesicles containing a chemical called a neurotransmitter fuse with the plasma membrane. The neurotransmitter molecules spill out into the synaptic cleft, diffuse across, and bind to the receptors on the receiving cell's membrane. The binding of neurotransmitters to receptors causes the attached ion channels to open. Depending on the type of neurotransmitter, receptors, and channels involved, this may excite or inhibit the receiving cell. Here we show excitation. Sodium ions rush in, changing the polarity of the membrane, and an action potential is triggered in the receiving cell. The neurotransmitter is broken down by an enzyme, and the ion channels close. Enzymatic breakdown is one of the several mechanisms by which transmission is terminated at chemical synapses.
So that animation is kind of nice. It shows most of the details that you do need to know. So just to emphasize a couple of things, the message is, of course, coming from the presynaptic neuron. It's these chemicals that eventually have to be released into the synaptic cleft. They need to bind to a receptor. And in this case, when they bind to the receptor, what it did is it opened up a channel. So this channel is very, very specific. This channel was specifically for sodium. We know that there's more sodium on the outside of the cell, including the synaptic cleft, than on the inside. so the sodium rushes in. And of course, positive charge is rushing in. That is depolarization, and that's why this would be an excitatory message. Now, there are a few other details that are involved as well, and for that, what I'm going to show you is another animation here. This one is at a neuromuscular junction, and you should know that term as well, neuromuscular junction. And all it means is a junction between a neuron and, in this case, a muscle. Events are pretty much the same, and we can see in the picture here that we do have the axon terminal. We have the synaptic vesicles containing the neurotransmitter. We do have the receptor on the postsynaptic membrane, but we have something else here as well. We do also have this green channel here. This green channel, not a major point for you, but it is a calcium channel, and the little green circles, those are representing the calcium ion. So when the message does reach the axon terminal, what we do want to have are these vesicles moving toward and fusing with the cell membrane, releasing their contents through the process of exocytosis. But something needs to initiate this, and what initiates this is calcium that's rushing in. So when the action potential reaches the axon terminal, it opens up calcium channels, calcium rushes in, and that leads to the release of the neurotransmitter. What we're going to see is that after we do get a response in the muscle at the neuromuscular junction, after that, something else is going to happen with the neurotransmitter, and we'll pay particular attention to that and also to the name of the neurotransmitter in this case, which is a name that you do need to know.
An action potential arrives at the presynaptic terminal, causing voltage-gated calcium ion channels to open, increasing the calcium ion permeability of the presynaptic terminal cell membrane. Calcium ions enter the presynaptic terminal and cause vesicles to release their neurotransmitter acetylcholine from the synaptic vesicles into the presynaptic cleft. Diffusion of acetylcholine across the synaptic cleft and binding of acetylcholine to acetylcholine receptors on the postsynaptic muscle fiber membrane causes an increase in the permeability of ligand-gated sodium ion channels. The movement of sodium ions into the muscle cell results in depolarization of the postsynaptic membrane. Once threshold has been reached, a postsynaptic action potential is generated and is propagated over the muscle cell membrane. Acetylcholine is rapidly broken down to acetic acid and choline in the synaptic cleft by the enzyme acetylcholinesterase. The choline is reabsorbed by the presynaptic terminal and combined with acetic acid to form more acetylcholine, which enters the synaptic vesicles.
So many of the events were similar to what we saw with the previous animation. Again, the couple of things that I will point out, you do need to know the specific neurotransmitter that is released that's going to target a muscle, and that neurotransmitter is called acetylcholine or acetylcholine. It is released into the synaptic cleft. As they said, it binds to a receptor, opening up sodium channels. In this case, the opening up of sodium channels and depolarization, because it is a muscle, it will result in the contraction of the muscle. But now you need to have that neurotransmitter released so the muscle can respond to subsequent messages. So what it's showing in blue here is an enzyme that's going to break down the neurotransmitter acetylcholine. It no longer fits in the receptor. It's released from the receptor. So here we see that it's broken down into acetic acid and choline. There's the name of the enzyme. Either goes by the name cholinesterase or acetylcholinesterase as it's shown here. And yes, you do need to know that name as well. And then eventually, the acetic acid and the choline, it's going to be transported back into the presynaptic vesicles, packaged up again, and it's just going to be recycled. So those are some additional steps that we do see with this animation here at the neuromuscular junction. So the steps that are involved, we have voltage-gated calcium channels again, which is going to initiate the movement of the synaptic vesicles toward the cell membrane, fusing with the cell membrane through the process of exocytosis and releasing the contents, the neurotransmitter, into the synaptic cleft. From there, very, very small space, so the neurotransmitter can diffuse across the synaptic cleft. It'll bind to a receptor on the postsynaptic cell membrane, and then we'll get a response. Again, that response, it could vary. It could either be the opening of sodium channels, or it could be the opening of some other channels, leading to, instead of depolarization, hyperpolarization. And finally, we do need to remove that neurotransmitter. So in the case of the acetylcholine, it is broken down by acetylcholinesterase or cholinesterase, and we do have the reuptake and the repackaging into the presynaptic vesicles. So this point here at the top, this is the one that we have been seeing, where we're opening up sodium channels, leading to the depolarization. The name for this, because it is excitatory, is it is called an excitatory post-synaptic potential, or EPSP. Let's just take a look at another quick little animation of this. So message coming down through the axon terminal, neurotransmitter released, binds to a receptor on the postsynaptic membrane. That's going to open up a channel, in this case the sodium channels. Sodium rushes in, we get depolarization, and if we get enough depolarization, then an action potential is going to be generated in the postsynaptic membrane. But that's not the only possibility. Again, we can have an inhibitory message. Now remember that there's more potassium inside than outside. So if we open up the potassium channels, then we're going to get hyperpolarization rather than the depolarization. So let's take a look at this one here that's showing the inhibitory message. Neurotransmitter released. Here we're going to have two different kinds of channels. We're going to have a potassium channel that we see on the left. Potassium rushes out. And chloride, which is negatively charged, there's more of it on the outside. It is going to rush in, and that's going to cause even more depolarization. Let's just play that one again. Neurotransmitter released, or more than one neurotransmitter, one binding to the potassium channels, the other one's binding to the chloride channels. Potassium rushing out, chloride rushing in, and that would lead to the inside becoming more negative. That would make it more difficult for the postsynaptic membrane to generate a message, and that's why it's called inhibitory. And just one final clip I'll show you here.
Communication between neurons and communication between neurons and muscle occurs at specialized junctions called synapses. The most common type of synapse is the chemical synapse. Here, we examine the events that take place at the neuromuscular junction, a chemical synapse using the neurotransmitter acetylcholine. Synaptic transmission begins when the nerve impulse reaches the presynaptic axon terminal. Depolarization of the presynaptic membrane initiates the sequence of events leading to transmitter release and activation of receptors on the postsynaptic membrane. Shown here are the principal molecules and organelles necessary for release of neurotransmitter. Synaptic vesicles exist in distinct pools, either tethered to the cytoskeleton in a reserve pool or free in the cytoplasm. Some of the free vesicles make their way to the plasma membrane and dock as a series of priming reactions prepares the vesicular and plasma membranes for fusion. The membranes of the synaptic vesicles are drawn together via protein complexes, collectively called snares, that are expressed on the vesicle and presynaptic membrane. When the axon terminal is depolarized, voltage-gated calcium channels open, and calcium ions rush into the axon terminal. Some of the calcium ions bind to a protein on the synaptic vesicle membrane called synaptotagmin. When calcium binds to synaptotagmin on the synaptic vesicles nearest the active zone, the vesicles are drawn even closer to the presynaptic membrane. The vesicles fuse with the axon terminal membrane and release their transmitter cargo into the synaptic cleft. Some of the transmitter molecules bind membrane. The response of the postsynaptic cell, either excitation or inhibition, depends upon the particular neurotransmitter and receptor combination. For example, the receptor for acetylcholine is permeable to sodium. After binding acetylcholine, the channel opens and sodium ions enter the postsynaptic cell, thereby generating an excitatory postsynaptic response. Transmitters are inactivated or removed rapidly from the synaptic cleft, so that transmission is brief and accurately follows the presynaptic input signal. For acetylcholine, an enzyme in the synaptic cleft, acetylcholinesterase, breaks down acetylcholine into choline and acetate. The release of transmitter from the receptors causes the channels to close. Not all transmitters are broken down by enzymes in the synaptic cleft. Many transmitters are rapidly cleared from the synaptic cleft by being taken up into the presynaptic terminal by special proteins called transporters. This process is known as reuptake. Reuptake not only cuts off the synaptic activity promptly, but also allows the terminal to recycle transmitter molecules. The membrane needed for creating synaptic vesicles is also recycled via endocytosis of the presynaptic membrane. The recycled vesicles are refilled with neurotransmitter molecules and are ready for another round of synaptic transmission.
So how is the decision then made by a neuron as to whether or not a message is going to be sent down the length of that neuron? Let's just go back here to one of the previous slides way at the beginning. What it really comes down to is, well, a little bit of math. So we had two different kinds of messages impinging on our motor neuron. We had our excitatory messages, excitatory interneurons, and those are the ones that are going to open up sodium channels, allowing sodium to rush in and leading to depolarization. The opposite, of course, are the inhibitory messages, which are going to now open up possibly potassium channels. Potassium would then rush out, and that would lead to hyperpolarization. In order for this neuron to generate an action potential, though, what we'd then want to have is more of the excitatory ones than the inhibitory ones. So let's say, for example, that there are 100 interneurons that are excitatory and they are opening up sodium channels at the dendrite end of this motor neuron. And let's say that there are 10 of the messages that are inhibitory. So adding these together, we can see that there are many, many more excitatory messages than inhibitory messages. And if there are a sufficient number to reach that magical threshold of somewhere around minus 30 to minus 40 millivolts, then that's going to be sufficient to generate an action potential, and then a message will be sent down along the length of the neuron. So adding together the excitatory and the inhibitory messages, this is simply referred to as summation. So if we drag back down to that slide, where it does talk about summation, so it's referred to as summation of the postsynaptic potentials. So after the synapse, the potentials, a bunch of them inhibitory, some excitatory, and depending upon, again, what you have more of, typically determines whether or not the neuron will generate and send an action potential. A few more kind of important pictures that we'll take a look at here. So what it's showing at the top, the yellow cells are representing the motor neurons, and we can see in this picture here that we do have some messages that are going to be inhibitory. So the eye indicating that these ones are going to be inhibitory, and we have other ones that are going to be excitatory. So excitatory ones they have labeled as E1 and E2. Now if we take a look at each of these pictures in sequence, the one on the left hand side, what we can see is that we do have an excitatory signal. So because it is excitatory, we will have depolarization that's taking place, but notice that we're not quite reaching the threshold potential. So again, it's going to vary from neuron to neuron as to exactly what that threshold potential is going to be. In your textbook, I think the number is somewhere around minus 55. Just prior to this, I think I mentioned minus 35. So it will vary a little bit from neuron to neuron. At either rate, though, what we do see here is that the threshold, it is not reached. So if there's not enough of a stimulation, not enough depolarization to reach the threshold, what we can see is that, well, the neuron just goes back down to the resting state of about minus 70 millivolts. If we take a look at the next picture, though, we can see now we don't just have one excitatory stimuli, but we have another one immediately after it. So in this case here, those two signals, they are going to add together, one on top of the other. And because now we have two excitatory and none of the inhibitory, that is enough to now reach the level of the threshold potential. Once that happens, now a whole bunch of sodium channels are going to open up, and enough will open up to lead to an action potential at the dendrite end. The third one that we're taking a look at here, now this is still summation. It just gives it a slightly different name, a spatial summation. In this case, it's not one excitatory after the other one, but it's both of them at the same time. It turns out achieving the same result, though. They are added together, enough excitation to reach the threshold potential, and again, as long as we do that, we get an action potential. The last one here is the only one where we do see inhibitory. So we can see if it is inhibitory, yes, we do in fact get the opposite of depolarization, we do get hyperpolarization. And finally, this one right here, what happens if we get an approximately equal number of excitatory and inhibitory messages? Well, it doesn't really change, and it stays right around the original resting membrane potential, which again, for most neurons, is typically around minus 70 millivolts. And this slide here is just showing a sampling of some of the neurotransmitters that are involved in transmitting a message from one neuron to another one. The only one that you are really responsible for knowing is the first one here. You do need to know the name, acetylcholine. You should know that, at least at the neuromuscular junction, acetylcholine is an excitatory neurotransmitter. In other parts of the nervous system, it may have either excitatory or inhibitory effects, but at the neuromuscular junction, it is typically going to be excitatory. And I give you just a few other neurotransmitters. We will come across these. We'll briefly talk about these, but these are the ones you don't need to memorize. And what that means is that if you are asked a question by me or on the diploma exam, you will be given a context box to provide you with some information about these neurotransmitters. Now, a couple of these I do have on your assignment sheet. This one here, GABA, or GABA aminobutyric acid. This one is typically considered an inhibitory neurotransmitter. And the next one, glutamate, is one that is typically considered to be an excitatory neurotransmitter. But quite often there are questions where they are talking about the neurotransmitters dopamine and serotonin as well.